Get started with our next presentation, which is being co-presented by Nick Rains and Len Balick. Nick is the environmental coordinator for the Troy Mine with Hecla, Montana. Nick received a bachelor's degree in mechanical engineering from Wright State University and spent the first several years of his career as part of a research and development team in the aerospace industry. He moved to Montana from the Midwest in early 2005 to pursue a career in environmental engineering. Upon arriving in Libby, he spent 7 years with a consulting firm, conducting and overseeing investigation, design, and remediation activities at a number of U.S. and international hazardous waste sites. He then spent 5 years focusing on environmental health, sanitation, and public health with the Lincoln County Health Department. Nick joined Hecla, Montana team in late 2007, and he oversees reclamation of the Troy Mine and permitting of the Montanore and Rock Creek projects. Leonard Balick is with Herrera Environmental Consultants. Leonard, he is a senior ecologist with over 35 years of experience in the field. He worked for 16 seasons for the U.S. Forest Service as a technician with forest inventory, habitat typing, reforestation, and other forest management duties. In '87, Len helped found a native plant nursery to provide native plant restoration services for revegetation of mines and other disturbed sites throughout the West. His primary focus area with Herrera is erosion control, habitat enhancement, and exotic vegetation control. He continues to work on the investigation of innovative restoration techniques for establishing native plants in arid regions. So welcome on to Nick and Len, and thank you very much for being with us today.
Thank you, Susie. Thank you. Can you hear my audio okay?
Yes, you're good.
Excellent. Presentation visible?
Yes.
Perfect. Well, thank you, Susie, and everyone else who has organized the conference. I know a lot of work went into it, especially adjusting last minute, and definitely turned out to be a very successful event. And thank you for the opportunity for Hecla, Montana to present as well. The Troy Mine is a project we're very proud of, and very appreciative to be able to share our progress and work that we've been doing out there. Just a quick check, Len, is your audio working as well?
Yes, it is.
Excellent, thank you.
Yeah.
Okay, so we'll dive in. A couple years ago, I think it was the 2018 conference, myself, Doug Stiles, our general manager, and Skip Leedy provided an update on progress at the Troy Mine at that time. So this will be kind of an update in addition to that previous presentation. So the Troy Mine is an underground silver copper mine operated in northwest Montana. It operated from the early 1980s through about 2015 when operations ceased. Construction started on the project in 1979 and operations began then in 1981. The tailings storage facility is a upstream construction tailings facility with sand fraction used for lift construction. It was started with a starter dike and then lifts of tailings built up from there. As I said, construction started in 1979. It started with harvesting topsoil from the actual tailings footprint and placing it adjacent to the tailings footprint to the west and building up lifts from there. During that placement of topsoil, harvesting and placement of topsoil for reclamation use later, we also ended up with some depression areas between the topsoil stockpile areas and the tailings facility, which ultimately became what we refer to as the tow ponds, or tow ponds adjacent to the TSF. At the cessation of mining operations, we ended with a very rough estimate of about 43 million tons of tailings in the TSF, or tailing storage facility. We're very fortunate. The tailings themselves are a benign material. It's a quartzite sand with very low levels of residual metals. So as we ceased operations in 2015 and began to look forward to reclamation of the full Troy mine site, our focus initially was on the tailings storage facility. We began full reclamation on the TSF in 2017 and completed the vast majority of all reclamation in 2019. We started out with a couple key goals in our reclamation. One was to reestablish and improve wildlife habitat. You can see in the photo down there, this is a shot from up on a nearby peak looking down on the tailings facility with the Cabinet Mountain Wilderness in the background. The TSF lies between the Cabinet Mountain Wilderness or Cabinet Mountains and the West Cabinets or proposed Scotchman Peaks Wilderness. So one of the key goals was to reestablish this or improve wildlife habitat in this area. We wanted to just reclaim disturbed lands and return them to a comparable utility as the surrounding lands, increase the habitat diversity beyond the proposed in the original reclamation plan, and provide wildlife security consistent with National Forest objectives, Kootenai National Forest objectives. In addition to that, we looked to protect air quality in the surrounding area by eliminating dust that could potentially come off of the tailings themselves. Ultimately, to summarize this, our whole goal was to reestablish vegetation and return this area, the TSF, to the climax forest, essentially a surrounding forested area, have it blend in with the surrounding areas. As I mentioned, the TSF reclamation was broken into essentially two distinct phases, a couple sub-phases, and completed over 3 field seasons. On the right, you can see an overview of the tailings facility looking down on top of it, plan view. One thing I forgot to mention is the tailings or TSF are broken into 4 distinct cells, starting on the south end with cell 1 and moving sequentially 2, 3, and 4 to the north, and that was based on operations. During those 2 distinct phases and then multiple sub-phases, we broke up the actual reclamation activity over 3 field seasons. The reclamation plan called for essentially capping or covering the tailings facility with soil and revegetating. So our approach to that reclamation was to rip and regrade the tailings surface itself. So initially rip and regrade. There were multiple dikes dividing these cells that would be regraded out. Some uneven and erosion areas on the tailings that were regraded out and ripped. We then harvested the topsoil, our cover soil, which I mentioned earlier, was stockpiled on the west side of the tailings facility. I'm not sure if my pointer will actually show up, but along the west side there you can see the tow ponds, and then just west of the tow ponds were the topsoil stockpile areas. We then placed subsoil material or borrow material on the surface of the tailings. We then placed a cover soil. Due to compaction during that placement, we would then rip the surface of the cover soil in order to allow for better seed bed for seeding and planting. We would then seed with native grasses, which Len will get into a discussion on more detail here in just a bit. We would also then plant seedlings. And then another big piece of this project was to construct some stormwater management features along the west side of the tailings facility. That's kind of what you can see down in the bottom left-hand corner of the plan view figure there. Some stormwater catch basins for managing stormwater off of the embankments of the TSF, as well as stormwater in the tow ponds and adjacent topsoil stockpile areas. So the TSF reclamation completed so far. As I mentioned, the vast majority, virtually all of the reclamation is complete on the TSF. That included placing cover soil over 290 acres of the TSF surface. We placed over 500,000 bank cubic yards of soil. We then seeded 354 acres. That includes the surface of the TSF, as well as the topsoil stockpile areas, reclaimed access roads, areas adjacent to the stormwater management basins, other areas surrounding the footprint of the TSF. We've planted over 185,000 seedlings. And in addition to this, which Len will also touch on, is we transplanted about 136 medium-sized trees from the topsoil stockpile areas out onto the tailing surface after soil was placed. In addition, as I mentioned, we created the stormwater management features along the western side of the TSF. Couple quick photos here just to show the progress. This was pre-reclamation, August 2016. The tailing storage facility, as you can see, exposed tailings on the surface. At that time, we were still placing water on the surface for irrigation and dust suppression, essentially. 2017, this was following our first year of reclamation activity. The lower portion or southern portion in this photo shows cells 1 and 2 with cover soil placed. This is late 2018 after the completion of phase 1 and then our first round of phase 2 cover soil placement and revegetation. This is late in the year, so a lot of the vegetation was browning up for the year. But you can see at this point, the vast majority of the tailings had been covered with cover soil and large portions had already been revegetated. One thing I forgot to mention is that kind of in the center of this picture, center of the tailings, you can see a small area. It's just under 10 acres. I mentioned we've completed virtually all of the tailings reclamation other than this 10-acre spot that we are leaving open for reclamation as we move up to the mine and mill facilities. In July 2019, you can see here we're starting to really see some good growth on the areas revegetation on the southern portion, so phase 1. That's cell 1 and 2 you can see on the bottom of the photo, and then also on the northern portion, cell 4, way up in the background of the photo there. These areas were all planted at this point. They've had multiple seasons for regrowth, and we're starting to see very solid growth on all of the native grasses as well as seedlings that were planted. This is a photo from June of this year, early in the season, so we weren't starting to see everything green up yet. But again, you can see continued growth on those portions that have had multiple growing seasons. The the portion in the center of this picture, cell 3, was the most recently reclaimed area that was covered in 2019 and then received revegetation in late 2019. So it's at this point was just going into its first growing season. You also note on this picture in cell 1 and 2 there, the southern or lower portion, lower left-hand portion of the picture, you can see some areas that are showing less growth. Those are areas that we continue to monitor, as Len will talk about with the revegetation monitoring, and have continued to look at options for amendment and fertilizing, which I'll get into in just a little bit here. So talk a little bit about our reclamation design and how we ended up where we were. The original reclamation design for the Troy Mine Tailing Storage Facility called for an 18-inch cover of soil placed in one lift over the entire surface of the tailings. As we started looking at the actual amount or volume of cover soil available in the topsoil stockpile areas, we started to realize that there was not a sufficient volume of material to do a full 18-inch cover, and started to think about some alternatives. In addition to that, or I guess a reason for that limited volume of cover soil, was that, as I mentioned, the west side of the TSF is flanked by a series of tow ponds. Those tow ponds were not initially part of the TSF design, but as the topsoil was stockpiled, they naturally formed depressions and filled in over time. At some point during operations, those tow ponds were identified as habitat for western toad, which is a species of concern in Montana. And in order to preserve that habitat, the final reclamation design included leaving those tow ponds in place. In order to leave those tow ponds in place, we had to look at stability of the embankments around the tow ponds, which reduced the overall volume available for cover soil. So in addition to looking at the cover soil availability, the amount, we then went back and looked at a couple other aspects of cover soil design, and that would be metals uptake through the soils and into vegetation, final vegetation on the tailing surface, soil utility, so ability to regrow vegetation similar to the surrounding area, and also looked at the pre-TSF construction soil conditions. What was the soil like in the area prior to construction? From the metals uptake perspective, we did some test pitting around the TSF in the embankments where trees and vegetation has been growing for 20 plus years. We also collected samples of the vegetative material on the surface and compared metals in those vegetation samples to standards set for several different species of receptors. That would include meadow vole and deer. What we found is that there was no difference in shallow cover, so 6 to 8 inches of cover soil, no difference between that and larger lift of cover soil up to 18 inches of cover soil. What we saw was no difference, statistically no difference between the two when it came to a potential toxicity to those receptors. All of the results were well below no observable effects limits for both deer and vole, which you can see in those figures down below here on the bottom of the slide. In addition to that, we saw soil utility, that there was virtually no difference, no statistical difference between 8 inches of cover soil and 18 inches of cover soil as far as regrowth potential on the tailings. That combined with some research that we did looking back to historic documentation of the pre-construction tailings, or prior to construction of the tailings storage facility, showed that topsoil in the area, in the footprint of the TSF, topsoil depth varied greatly over the entire footprint of the TSF. So all of these factors combined, we worked very closely with Montana DEQ Hard Rock to propose a change in our cover soil depth, and with DEQ's support, ended up with what was referred to as the mosaic alternative. This basically gave us a reclamation design that varied the depth of cover soil over the entire TSF, anywhere from 8 inches of cover soil up to ultimately almost 24 inches on the north end of cover soil and borrow material. This more closely mimicked the original conditions of varied cover soil depth across the TSF, and will provide us with the same results for revegetation as we would have seen with that single 18-inch lift. In addition to that, part of our reclamation design change included placing the soil in multiple lifts. As most of you would be familiar with construction, placing a single lift of 18 inches of topsoil or any soil material and getting a consistent depth across that is a construction challenge, to put it lightly. So under the new design, we were able to put it in multiple lifts. Last thing, as I mentioned, you saw on the slide, a few slides back, some areas that were showing limited growth. So what we have done is we have looked at and utilized a variety of soil amendments and fertilizers throughout reclamation. Two of the key fertilizers and amendments that we have used are Biosol Forte and a combination of Proganics, BSM, and Richlawn. We've used these two at different times to test out their ability, how quick they respond, how well the growth shows year one versus multiple years after. What we found is both of these options have worked out really well. You can see the two pictures, August 2019 and June 2020, that is cell 4 on the north end. Same with the other picture there. This is an area where we used a combination of Proganics, BSM, and Richlawn. Other areas on the TSF we have been amending with a Biosol Forte. What we've done is as we've placed soil on the TSF, we have continually monitored the soil characteristics, things like water holding capacity and available nitrogen. When we see areas with lower available nitrogen, we have evaluated and applied amendment and fertilizer to ensure that we get good germination and good regrowth as quickly as possible. As I said, we've found good success with both of these amendments. A couple of things that I've noted here: Biosol Forte, which is a fermented plant-based organic fertilizer. It provides a slow release and long-lasting nitrogen source. It is a pellet format that we use, and it's relatively inexpensive to apply. It can be broadcast spread, either machine or hand broadcast spreaders. Proganics, BSM, and Richlawn combination that we've used Proganics is a recycled refined bark and wood fibers blended with a number of different things, biopolymers, biochar, seaweed. And Richlawn is a organic poultry-based fertilizer. That combination provided very, very good first year growth. I believe it's a slightly quicker release nitrogen source. As I mentioned, great first year response. However, the one challenge is it does have to be hydromulch applied and is therefore slightly more expensive to apply. But we are seeing excellent response and excellent growth in all areas that we're applying amendment to. With that, I'm going to turn it over to Len for a little discussion on the actual revegetation activities.
Good morning. First off, I'd like to thank Hecla for letting me help, letting Herrera help on this project. It's been great. We've been able to work the revegetation right from planning on through implementation, which is great. A lot of times we end up with planning and then it goes on to someone else that takes care of the applications, which may or may not work well. And other times we just have portions. So this was great to have that, and I really appreciate it. This revegetation plan that we put together, that Herrera put together, was based on the native vegetation of the site and was broken up into upland forest, moist forest, grassland, and shrub zones that we based on microtopography and soil types. Next slide, please. Nick?
I just changed it.
Okay, thank you. So the site was, like Nick said, was salvaged and saved from the original construction of the facility. And it was ripped, and it was also left in projects that looked like someone's planting a lawn. In this case, you can see the rough conditions of the site, which really facilitates a better revegetation success. And go on to the next one, please. Nick mentioned the trees that were growing on the topsoil stockpiles. And this is something that I kind of noticed and thought, well, that'd be really good the first thought was, well, we'll use it as woody debris to spread on the site. And then I did a little bit more research, and we used on the textured soils that were good enough to be able to harvest those. You can see the photo on the bottom right is a temporary nursery. We tree spaded those, wrapped them and made them in ball and burlap, put them in a temporary nursery, covered them with wood chips, and it was great because during construction, the truck driver and he watered those trees throughout the whole first construction season, which really resulted in good survival of those trees. Next slide, please. We then planted those salvage trees in pods across the site in different places.
Hey, Len.
Yes.
Len, I'm sorry to interrupt. This is Susie. Your sound is coming in kind of spotty. Can you get a little closer to your Wi-Fi router, maybe?
Okay. Can you hear me okay now?
Yeah, you sound really good now.
Okay. We put planting these trees on site. It was kind of interesting. We took an approach that was different than typical landscaping. Typical landscape trees, you plant them, you put big poles out around it and guy wires to hold the trees up. This site is extremely windy, so what we did is we used tree anchors that anchored the root balls instead of using those top poles. That way there was no anchor ropes or other poles to have to be maintained. And we got good survival of those trees, which gave us some really nice habitat. Next slide, please. So it's really important to have site-adapted seed and site-adapted plants for these projects. So what we did is we purchased seed from Northwest Montana and Northern Idaho seed sources for the seeding of the shrubs and, seeding of the grasses and forbs. And also we did a major seed collection for all the woody plants, the shrubs and trees that were to be grown. We used a local nursery to grow those trees, and what we did was to ensure that we had good quality trees. We made periodic inspections of the nursery, the quality of the trees, so when the trees arrived on site, they were what we expected. The seed mixes were developed again for those different habitat types, and we seeded with a Brilliant seeder, kept track of all the seed that was put down and monitored that really carefully. We had a person out there on site that would check out a site that had been seeded, GPS that, and check to make sure that we had the proper rates for the seeding. The tree and shrub planting, we also hired a local Western Montana contractor. It's interesting because a lot of these people that worked on this crew and the owner of this crew, this plan, actually worked with me when I was at the Native Plant Nursery back in the 80s. Real good planters, professionals. They were out planting and doing a really good job. You notice woody debris on the right-hand side. Those folks were really good about getting in and planting trees and shrubs in there where they would have shade and where the snow would stay in the wintertime and allow those trees to survive better. The nursery on the left was taken care of. We checked that nursery daily for health of the trees and shrubs. Next slide. One of the things that we started with right away before we ever did anything beyond just the original revegetation planning was we started doing weed surveys. And so a crew would go out every year, walk even the access roads and the site to identify potential weed problems. We would GPS all those sites and pass that information on to weed control specialists that could treat those. And then we go back the next year to make sure that they were followed up with. Back of that last photo, you could see some of the weeds that we had, which were we had some oxeye daisy, we had a lot of clover, which really isn't a noxious weed, and then at the upper left there we had a little tiny bit of meadow knapweed that we got rid of, and St. John's wort, and that's been taken care of also. We'll continue that weed control or weed surveys on into the future. Okay. The monitoring, what we're doing for revegetation monitoring is we're putting in permanent plots. As soon as an area has tree and shrubs planted, we'll go out and put in permanent steel plots with a plastic cap, GPS where that plot is, and then we have two plots. One is a small radius plot that we measure the number of seedlings of grasses and seeded forbs that come in, and then a larger one at that same place, larger radius, that will where we identified all the trees and shrubs that were originally planted in there. We can go back and see which of those survived and if some of those are spreading, and it's actually doing very well. So the results of this project, I think, are excellent. I'm just really excited about it. The results are kind of based on the soils and so on. So there's some spots that we know are not doing as well, but we can identify those quickly. By having this constant monitoring, we can take action, like Nick said, is adding other amendments or other treatments. We can also see if we're having trouble with browsing, which is amazingly low compared to the amount of deer and elk that we see on that site. I think the revegetation on this site is a demonstration how careful planning, seed and plant procurement, and oversight, along with ongoing monitoring, can really result in quality vegetation establishment. And with that, I'll turn it back to Nick.
Thank you, Len. So I know we're getting short on time, so I'll try to run through the last few slides here really quickly. Another key piece of our ongoing monitoring at the TSF is ensuring that water quality and aquatic life in adjacent streams continues to thrive. Some of the monitoring that we do over the last 35 years, we have looked at both water quality and aquatic life in adjacent streams like Lake Creek, which is immediately west of the TSF. Over those 35 years, our data collection has shown that metals concentrations in Lake Creek, copper and lead in particular, are well below Montana aquatic life standards. Copper and lead concentrations in Lake Creek are also consistent between locations, so above or upstream of the TSF, as well as adjacent to or downstream of the TSF. We've looked very closely at metals intolerant macroinvertebrates in Lake Creek and other surrounding streams. Over those 35 years, we've shown that our monitoring indicates no difference in Lake Creek above or below, upstream or downstream of the TSF, and no significant changes in the macroinvertebrate communities over that 35-year monitoring period. In addition, I mentioned one of our key goals was to improve wildlife habitat. The TSF sits in an area where it provides essentially a vital wildlife corridor between the Cabinet Mountains and the West Cabinets. We have continued to see wildlife accessing through the TSF on a consistent basis and increasing as reclamation has been completed. All of the photos that you'll see in the next few slides were taken directly at the TSF. We regularly see moose, elk, deer traveling through that corridor and near and around the TSF. Number of different bird species, ducks, loons, geese, And we have quite a healthy bear population that makes its way through. A couple more photos here taken on the TSF over the last year and a half. One other thing is the western toad. As I mentioned, this is a species of concern listed by Montana Fish, Wildlife, and Parks. They're listed due to declining population, I believe mostly due to a fungal infection. Once these were identified in the tow ponds, we took measures to ensure safe habitat during construction. This included the construction of what we refer to as the toad highway. The tow ponds during the season in which the western toad utilizes the tow ponds for mating and rearing of tadpoles and young during the spring and early summer. The tow ponds were surrounded by silt fence, and then essentially we created a corridor from the tow ponds out to Lake Creek. Ended up in several miles, between 2 and 3 miles of silt fence to provide this corridor and safe passage between the tow ponds and Lake Creek. Last, just wanted to mention a couple of the kind of lessons learned and applied throughout our reclamation project at the Troy Mine. One of the big things is key partnerships and coordination. We worked very closely with Montana DEQ and the Kootenai National Forest on the overall reclamation design and those changes to the design that I mentioned. Also a very key aspect, something that played heavily into the success of the TSF reclamation, is utilizing local and regional resources and expertise. As Len had mentioned, we worked closely with Herrera and then the Confederated Salish Kootenai tribes on re-veg plan, re-veg the seed collection, and actually installing, planting and seeding. A number of local contractors, everything from earthwork, dirtwork contractors to folks, engineering firms that we worked with. So on the dirtwork side, DG&S out of the Silver Valley, CDM Smith, Mall, Foster, and Alongi. A number of different folks have been involved in this project and have really provided valuable added value to its success. And then the other key piece is flexibility and adaptation. We, throughout the process, have had to adjust our process to ensure that we're getting adequate and appropriate cover depth, that we are monitoring revegetation and adjusting as needed for amendments and fertilizers, and just continuing to progress the site to that climax forest goal. With that, I will open it up to questions.
Okay, thank you, Nick and Len, for that great presentation. You do have a few questions here. The first is, what is your typical process for conducting your weed survey? I'm wondering, do you GPS points only or collect polygon data for high concentrations of the weeds?
So historically, during our weed surveys, and Len, please jump in if I misstate anything, Herrera has been conducting those weed surveys in the past, and we have typically done a combination, primarily GPS points for, I guess, higher intensity areas with more weeds identified. Those are then placed on a figure or map that shows what species were identified, where they were identified, the extent, an approximation of extent, lateral extent of that weed, and then that's passed on to a weed management a contractor, essentially a weed sprayer, who then goes out, verifies, and treats those weeds with a treatment that is typically developed both in combination with Troy Mine Inc., the weed management company, as well as input from Herrera.
Yeah, that's true. The point GPSs are usually the noxious weeds. We're really, a lot of it is we'll map out is just in areas, but we're getting down to where the noxious weeds are few enough to where they're just mostly points. And that really allows the weed control folks to go directly to those sites, and it allows us to track how well they've been doing.
And another part from that same commenter is, what are typical small, medium, high amounts, or do you count plants in representative plots or use another process?
For the monitoring, we put in permanent monitoring plots. So we don't do transects. What we do is, as soon as the trees and shrubs are planted, we put in a plot center and count all those trees that are within that plot. And for the trees and shrubs, then we have a smaller plot in the center of that, the smaller radius, that is for the grasses and forbs. So the first year when the trees and shrubs are planted, there's very few grass and forbs, so we probably don't sow anything in there. But then the next year we could see how much was grown within that center plot, which is the smaller plants, and we can see what kind of survival we had of the woody plants, the trees and shrubs in that wider plot.
Okay, and another question is, what kind of wind velocities do you expect across the top of the TSF? Do you expect, or have you observed snow drifts downwind from the tree pods?
We don't typically see any substantial snow drift. So I guess to answer the first part of that question, I don't have an estimate on wind speed estimates. We do see some strong winds at different times of the year. It is an open area. It's a valley between two significant mountain ranges. So we do see some substantial winds, typically coming out of the south heading northward. We don't typically see a lot of drifting snow due to the water content in our snow here. In the northwest corner of Montana, we get fairly wet, heavy snow. I often joke to people who haven't been up here before that if we get a snowstorm, it's usually still sitting on telephone lines and utility poles a week later because it's such a dense snow. So we don't see drifting in any sort of substantial amount anywhere on the site, and have not seen that or experienced that to date.
Okay, and also I lost a lot of trees to marmots at the TSF reclamation project in Wyoming. Any thoughts on marmots?
I've not seen any there. How about you, Nick?
No, we haven't. We do have some smaller, ground voles, other ground squirrels, and other things that we've seen on the embankments over the last 20 to 30 years. We have not seen any substantial losses, any tree losses yet along the embankments caused by, even when we see pioneer trees, naturally spreading trees along the embankments, we don't see a high rate at this point of loss due to any sort of significant browsing and or root damage from burrowing animals. We've been very fortunate.
And one last question before we move on. The agency's calculated a reclamation bond for the Troy mine with respect to the TSF based on your expenses. How accurate was that estimate, and what should have been done differently?
That's a very timely question, as we're currently reviewing that right now. We have found that after changes to the design so far, I would say the reclamation bond calculations have been pretty accurate. We've found some areas to be less expensive. Once we get into ongoing monitoring and, as I mentioned, the amendments to be placed, fertilizers to be placed, the long-term maintenance of the site, I think the initial or original calculations by DEQ have proved to be fairly accurate so far.
Okay, thank you.
Well, thank you very much for the opportunity to speak.
And you know what? We do have one final question that just came in. What is the status of the reclamation of the mine site, mill site?
Yeah, so just a really quick overview without getting too far into the weeds. We are, now that we have finished the tailing storage facility, we are making our way up the hill towards the mine and mill site for reclamation there. We are currently working on designs for underground water management, the final regrade and reclamation of the mine and mill site, adit closures, and then we'll proceed with construction as those designs are finalized and approved by the agencies.
Wonderful presentation. Any additional questions, please just email Nick and Lynn directly.
One thing, if I could chime in really quick to touch on that question about progress up to the mine and mill site. We have completed demolition of one of the large buildings at the mill site, the actual mill building, salvaged the ball mills and flotation circuits and moved them out, and then completed full demolition of the building itself. Then we're moving towards demolition of the remaining buildings in future seasons here.
Okay, thank you very much.
Thank you.
Okay, so we are moving on to our final presentation of the conference. And seated with me in studio is someone who needs no introduction, but I will introduce him anyway. He is one of our most charismatic presenters I think that we have ever had, and he is a longtime supporter of this conference. So without any more, we will turn it over to Rich Rogers with Bighorn Environmental.
Coming to you from Butte, Montana, where it's a smoky 70 degrees. They tell me, thank you. They tell me that they save the best for last, just like with the fireworks show. I think I heard that somewhere anyway. Well, I have a treat for you. It's on soil health. What is soil health and how do you measure it? What I learned investigating cover soils in a Superfund project. And as a special treat, what kind of soil structure do we find in young revegetation? I say it's a special treat because many times, not many times, but several times I've been at the conference when somebody asks a question about soil structure. Sometimes I think the answer is a little unsatisfactory, so we're going to actually look at it and you'll learn how to recognize some soil structure yourself. Okay, here we have a soil. What can you say about that? It's distinctive. You can see that it's different than the material just under it, much less the bedrock. And if you're experienced, you're probably looking at that bedrock saying, is that acid generating? Yes, it is. The soil is rocky, it's pretty thin, and it's dark. Why is that dark when the parent material is a yellowish color? That's coming from the carbon that accumulates over millennia in the soil, giving it that color. And that soil is supporting the plant community there. I see a lot of blue bunch wheatgrass. It's at 7,000 feet. I know there's Idaho fescue. You see mountain big sagebrush and off to the right. I see a blue penstemon plant over there. How can that generate, oh, that's going to be in the area of 1,000 pounds per acre per year there. How can it generate that? That's kind of what we're going to talk about today. I'm an autodidact. That means self-taught. The good news, I'm not a soil scientist. I don't claim to be, but I've worked with some of the best. Tom Keck, Don Strom, before that, Wes Larson. It's an apprenticeship, I think. Those guys are all expert mappers and classifiers, but I don't think so much on the microbiology end. So, an autodidact, what's the problem with being an autodidact? You don't know what you don't know. And what's the good part? Well, the good part is you're likely to not succumb to the latest fad, although some would say that soil health in itself is the latest fad. And these are some of the books that, this one here, actually surprisingly good, and a basic book for microbiology. So organic matter is the one thing that everybody's going to think of when we talk about soil health. And that's going to get all the attention. Maybe you're going to say your cover soil or your whatever amended soil has to have a certain amount of organic matter. That's the plant, animal, and microbial residues in various stages of decay. It represents the dominant source for microbial nutrition. But all OM is not created equal, and that's one thing we'll learn in detail today. Here's some more definitions. The organic fraction of the soil is a product of the synthetic and decomposing activities of the microflora. And we'll learn what microflora is too. It derives from plant constituents modified by the microflora and recalcitrant, and we're going to learn that too, microbial cells and metabolic products, like fecal pellets. One of the main things we have to know is that primary productivity drives the soil food web, and nutrient cycling drives primary productivity. Each depends upon the other. This is some vegetation right near Montana Tech, right near Fairmont, actually. I'm sure some of you are gasping, oh my God, he's got all this alfalfa growing out there. Okay, yeah, that's legitimate. Probably few of you have ever measured productivity, but if you'd like to take a guess, we're talking about 2 tons per acre per year. That's a lot. That's not irrigated. It's 2 tons per acre per year. And that is going to drive a lot of microbial activity. This is also near Fairmont, my revegetation. And this is more like 1 ton, maybe 1,800 pounds, 1 ton in there. There's some alfalfa, there's a lot more grass, and so on. Deer and elk and antelope love it. And this is the Clark Fork remediation project. So what we see here is, I don't have a pointer, but in your upper left, that's the cover soil, which is pretty sandy, and it's been amended with compost. And then in the center of the picture, they mixed it up with an excavator. Supposedly that microtopography confers some benefit. What do you think the productivity is there? Maybe about 300 pounds per acre per year. What do you think the soil food web is there? Not getting much to go on, and in a lot of places, they're not getting anything at all. I don't have a pointer for you, but right near that meter stick is some Artemisia decunculus tarragon on one of my transects. And on the same transect, yes, it's that big plant growing with the alfalfa. So the true miracle of nitrogen fixation, which is actually mediated by a bacteria, and we'll be talking about bacteria today. Here's one more. The microbial biomass in the soil is the driving force of most terrestrial ecosystems because it largely controls the rates of turnover and mineralization of organic substrates. So where do nutrients come from? They come from weathered minerals. They come from organic matter. 95% of the nitrogen and sulfur in most surface soils is in organic form. The percentage of phosphorus is less, but still represents the dominant form of nutrient supply. And this is from some data I have. We look at the amount in the soil and in the plant. Calcium, there's a lot in the soil. Well, that was a lime soil remediation project, and about 400. Magnesium, there's half as much and 3 times as much in the plants. Copper, yes, this is a contaminated soil, but only 8 parts per million in the plant. And zinc, much less, about 1/5 of what there is in 35 parts per million. Nutrients are released from the organic matter by bacteria and other soil microflora and microfauna. So these are words we have to get used to. Soil contains 5 major groups of organisms. Anytime you ask anybody that's an expert on soil health, they should be able to rattle off the 5 major groups right off. Bacteria, actinomycetes, fungi, algae, and protozoa. The first 4 are the microflora, and the last is microfauna. Now, I can't point with this, but this is the cytoplasmic membrane, and it says it enables some substances to pass in and out of the cell. I know the ones that are ahead of me in microbiology say, oh, the cytoplasmic membrane, that means a eukaryotic. But that's not why I put it up there. This is actually pretty important. The lower part is inside the cell, and the upper part is the soil matrix. Inside the cell needs carbon, it needs some nutrients, another carbons, in other words, energy, and it's where the enzymes are made. They pass out through whatever kind of membrane it is into the soil, where they join all the other enzymes that are out there. The ones from fungi, the other ones from other bacteria, the ones from the plants themselves, the ones that come from the protozoa. So it's quite a jungle, and they're all looking for things to degrade. Bacteria are losing their flagella and cilia, and fungi are losing some of their filaments, and all this is going on out there until these substances are brought down to simple level of just some sugars or something about that level that passes into the cell. Now, what passes into the cell may not be the same thing that was attacked by the enzyme that went out of the cell. So it's a big community of enzymes out there. This kind of summarizes that foodstuff must penetrate into microorganisms to be of use. But microbial cells are impermeable to many complex molecules. Like what? Like cellulose, for example, which first must be solubilized and simplified. This extracellular decomposition is done mainly by enzymes that are excreted by the microbial community. Elements and nutrients may be a mineralized or an immobilized state. I know this is a lot to try to take in, but I assume most people aren't well versed in it. Organically bound nutrients are immobilized. You are immobilized. If you're munching on a hamburger right now, that's immobilized. If you're eating ammonium nitrate, okay, that's mineralized, but I don't think you're eating that. The release of organically bound nutrients is known as mineralization. Mineralization is the inverse of immobilization. Now we've classified the organic matter. We can say it's labile. It's undergoing continuous biological breakdown. For example, simple sugars are quickly metabolized by many microorganisms. Here's a word we saw earlier, recalcitrant organic matter. Recalcitrant molecules are those that fail to be metabolized or mineralized at significant rates. The initial depolymerization is difficult and possible only by a few exoenzymes, and little energy may be released. But these become the key, these enzymes that can unlock the complex molecules. Each successive step can be affected by a wider array of microorganisms, exoenzymes. That's extracellular enzymes. Here's a little sidebar. Some organic matter is protected from degradation through microbial exclusion or clay organic matter complexation, so the turnover of soil of organic matter may be measured in hundreds or thousands of years. Bet you didn't know that. All organic matter isn't equally active in nutrient cycling. I don't have a cursor, so I won't be able to. I wanted to make this a quiz with an audience, but we're looking at typical forms of organic carbon in the soil. I wish I could get a feedback. Could anybody guess what's on the right? I seriously doubt it. It's, okay, I got one here that showed that on the right, it's aromatic carbon. And what's aromatic carbon? Well, it's carbon chains that are formed in a ring with conjugated double bonds, and it's very stable. Anything you find mainly in the soil is going to be in the recalcitrant. Not anything, but most of it is going to be in the recalcitrant category. And the next category, I do have them down on this one here. The next one is nitrogen-associated stuff, so you think about amino acids, nucleic acids, that kind of thing. Carbohydrates, you better believe they're going to be complicated ones. And fatty acid alkaline carbon, I don't know what it is, but there you have the stuff that's in the soil. And I wanted you to guess, how about what's entering the soil from plant residues? And this one I think you could do a lot better at. That big one on the right, that's cellulose. That second one, the orange one, that's hemicellulose. And the next one after that is some kind of carbohydrate, and then we have some smaller categories. Some proteins, okay, and some amino acid sugars. So what enters the soil is a lot different than what accumulates in the soil. And what you measure, when you just measure bulk soil, you're getting the recalcitrant stuff, and it's not really reflective of the part that's most active in nutrient cycling. So the composition, activity, and abundance of the microinhabitants differs greatly for humus. Now, some people use humus as a synonym for organic matter, but others use it for recalcitrant, and that's how we use it here, versus fresh, juicy substrate. I like this, a quote from Betsy Dyer, one of those books. To a bacterium, life on a soil particle is quite different than life on a plant rootlet. One's getting the exudates directly. Another very important thing to know, 2/3 of most fresh plant residues generally decompose in 1 year, the labile portion. 2/3 of the fresh residues decompose in a year. And these are contributed by root exudates and other. Now, I gave this talk to a class. This root exudates probably blows your mind a little. Yes, it's actually stuff coming out of the root into the soil. It costs the plant photosynthate. Why would it do that? That's how important it is to the plant to stimulate the microflora in the immediate vicinity of the roots. Also, excretory products sloughed off cells and root turnover. That means the roots that die every year. Whoops, my fault. About 2 to 5%, remember we just said 2/3, 2 to 5% of the carbon present in humus is mineralized each year, although the exact amount varies. So we have quite a difference between the fresh matter that's introduced into the soil and the stuff that accumulates in the soil, which is not very reactive. Here it is again, a major source of carbon and nutrients in the organic matter comes from root exudates. They generally represent about 5% of the carbon plants photoassimilate. So it's definitely a cost to the plant. What governs the rate of turnover? I just call them the usual suspects. Most of you could guess these. Temperature, water potential, redox potential, nutrients, porous pH, oxygen availability. There are going to be a lot of things that affect how fast things turn over. But nothing much happens in dry soil. Microbial ecology is aquatic ecology. And nothing much happens in frozen soil. And nothing much happens deep in the soil where there's little oxygen and even less exchange. And nothing much happens on the surface in an arid or semi-arid climate. First to dry, first to wet. First to freeze, first to thaw. It's a pretty tough environment right on the surface. And not much happens in saturated soil. But about 40% of field capacity is best. Alternating cycles of wet and drying, I spelled it wrong, promote activity. Well, this is the lesson that comes from wetlands. Wetlands stagnate, but when you have droughts and then a wetland, that's when you get a real flourishing of plants. There are active microorganisms, so active bacteria use whatever resources are supplied by the habitat. If resources are scarce, they may grow slowly and be hard to detect. And if you're interested in assaying the bacteria in the soil, that's important. And then there's dormancy. Dormant populations are opportunist, typically inactive, quiescent, awaiting an influx of resource, then flourish. They can erupt in lab cultures, so it makes it difficult to inventory soil bacteria through culture. Understanding what microorganisms are taking from their environment and depositing as waste is the key to understanding microbial ecology. And that we've gone over this, the attack of complex molecules limits the rate of decomposition, and most intermediates probably are broken down as quickly as they are produced. A brief detour, you can relax for a minute. As soon as you see the year 753, you think species Plantarum. Carl von Linné, the Swede who renamed himself in the Latin Carolus Linnaeus. 2 kingdoms. Life was simple back then. Plants and animals. What about the fungi? Surely they saw fungi. Yeah, well, they don't photosynthesize, and they're saprophytes or parasites, so let's put them in with the animals. R.H. Whitaker, whose virtues are still being discovered, significant figure, I think, 1969, among all the other work he's doing, he named 5 kingdoms. Plantae, Animalia. Fungi gets its own separate one, they absorb food. Protista, single cells such as protozoa, animals, and Monera. They lack a membrane-bound organelles and a nucleus. These are the prokaryotes. And when you see the prokaryotes, you're basically thinking bacteria. Then along came Carl Woese and Ralph Wolf, 6 kingdoms. They added archaea, very primitive bacteria, probably the first forms of life on Earth. And then came the 3 domains, archaea, bacteria, and all the stuff that you deal with almost every day, eukarya, the protista, fungi, plantae, and animalia. So bacteria are number one, at least in numbers. Number one, I mean in our soils. They're the most numerous and diverse organisms on Earth. Most are tiny rods, the rest are tiny spheres. They're all but identical. How you like that? So it's going to be pretty hard to identify them. Taxonomy is uncertain. They're not sexually reproducing. They look alike. Sometimes they function alike. And they've been described as a continuum of flowing genes. Horizontal gene transfer further muddies the water. Next to the Actinomycetes, they're filamentous bacteria. What does that tell you? Right off, it tells you that there's going to be a link between the fungi and the bacteria. It's not that clear. One of the best adapted to extreme water stress of the soil microorganisms, specialized in the degradation of some of the more recalcitrant polymers. Well, that's important. We know that's a bottleneck. They're active at high pH in compost production. They give compost that gray hue. And they often dominate in tropical and subtropical latitudes. Fungi, they're filamentous. They absorb food, that's key. They need oxygen. You won't find them in a wet soil. They tolerate acidity. They're saprophytic or parasitic. And they're parasitic mainly on plants. So for example, in Montana, most of your conifer parasites are going to be root fungi. Also, very importantly, the mycorrhizae, the root association that allows plants to get more nutrients. Algae, I don't think they mean algae. I think they mean the blue-green algae. They're really classified as bacteria. They're the cyanobacteria. Algae produce large amounts of extracellular polysaccharides. That means sticky stuff. They can act as soil aggregating agents. This can be important preventing soil erosion and structure stability of otherwise bare or poorly vegetated soils. You know what that is, macrobiotic crust. Some algae can carry out non-symbiotic nitrogen fixation using the enzyme nitrogenase. And just the last thing is the microphonae, small, often microscopic animals inhabiting the soil, single-cell protozoans, nematodes, those are unsegmented worms. The terrestrial forms are microscopic or darn close. Tardigrades, 8-legged arthropods, and the Protists kingdom. They inhabit water films. Well, we're back to needing water in the soil and pore spaces in plant litter and soil, feeding on smaller organisms. So Protist, if you remember, one of the 4 eukaryotic kingdoms, aquatic, mostly unicellular, no distinct organs, etc. Some algae included. On to what we're actually investigating. When would you measure soil health? Well, in a semi-arid climate, that would be the valleys in western Montana, when soil moisture is least limiting and photosynthetic activity is greatest. The microflora is most active at about 40% of field capacity. And my work was done in late May and June. Remember, we said that nothing's going to happen in a dry soil. You want to get there when they're active. And where would you measure it? Well, not in the bulk soil. At least that's what I think. The action's in the mycorrhizosphere and not too deep. The immediate vicinity of roots and hyphae that function as very fine roots. That'd be the mycorrhizae. I sample right underneath a large bunch grass, including the root fragments. I cut them off. They all go in the sample. The mycorrhizosphere is up to is a little more than, in my case, 2 decimeters. So you want to be pretty near the surface, right underneath the plant. Now, here's a picture from southwest Montana where I live. And anybody from around here is going to say, well, first thing, that gets good precipitation. how much? Minimum 16, probably 18 inches, something like that. Furthermore, looks like it gets a pretty good snow load too. And I see grasses and forbs, I see shrubs, and at the top there, a little left of center, I see a Doug fir limb. You could dig a hole anywhere here and you'd have the mycorrhizosphere. No worries about it. Here's another picture from southwest Montana. This is out by Bannack. And Bannack was the first state capital, and they overgrazed the heck out of it. The topsoil's all gone. And if you sampled in just anywhere here, it's human nature, guaranteed, you're going to pick an empty spot. It's just what everybody does. To get something on plant health here, you're going to have to go right under one of those small sage brushes with the grasses under it to get a zone of activity. Just for reference, that's a picture from Silverbow Creek, which is the main topic here. The project, Silverbow Creek, super fund, remove fluvial mine waste, bring in deep borrow to serve as cover soil. These samples were 1 to 15 years old, so I have time as one of the variables. That's mine waste. We all know what that looks like. That's the stuff that's removed. That's cover soil. Very funny. I took this picture. I was asked, I hardly ever go to a bar area. You see all that stuff in the middle of the picture? It looks like some kind of aggregates. I didn't notice it when I took it. You look higher in there and you see what you expect cover soil to look like. In this case, it's a sandy loam, and that's exactly what it looks like. So a bit of a mystery why we have something that looks like structure on there. How to measure soil health? Well, the first thing is, if you have revegetation that looks like this, and this is near Fairmont too, I'd say don't worry. You're getting, if that's 10 years old and your revegetation looks like that, trust me, you have healthy soil. You have nutrient cycling going on. Or we could look at some soil animals and say, well, if we have soil animals, yeah, we probably have some measure of soil health there. But how would we more directly and quantitatively measure soil health? There's 3 main ways that I know of. One I call the smorgasbord, technically called bioplates. This is a really great method, and if any of you want to hire me to do it, I'd be happy to. What you do is you get a bunch of substrates. A substrate means a food source. What are they going to be? Well, you're going to go right back to those pie charts that we had. And you're, on the one hand, you're going to have hemicellulose, cellulose, and proteins and fatty acids. And you're going to go to the other one, you're going to say we have to have some aromatic carbon and so on and so forth. And maybe get about 2 dozen different substrates. You have some amino acids in there. And you get a pinch of this soil and put it on these substrates and see what happens. Are the microorganisms capable of degrading that substrate? You're probably going to record it in something like, vigorous, moderate, low, none, something like that. You need a principal component analysis to evaluate it that way. But anyway, that to me is the kind of the ideal. How many substrates can this community degrade? The second one is cultural, it means culturing, taxonomy, 5 major groups that we already named: bacteria, actinomycetes, fungi, algae, and protozoa. Taxonomy is not so easy. For example, I said this, a single species of fungus can grow into forms that bear no resemblance whatsoever. But we already talked about the bacteria all looking like tiny rods and tiny spheres. So this is for the pros. This is for people who have looked through a microscope a lot and know what they looked at. Even so, there's problems. This is what Betsy said about that. Do these cultures represent the normal active biota? Of ordinary or normal biota are far more difficult to enumerate and evaluate due to their cryptic habitats, slow growth, and whatever, and subtle activities. So now an interesting side note on this. This is the exact approach I took when I evaluated the effectiveness of compost on this same project, which I can tell you right now was not much of a benefit at all. And the lab that I used was BBC Labs in Tempe, Arizona. Went out of business only a year or 2 later. Now I don't have any real insight on why they went out of business, but it's interesting that just as interest in soil health was building, that one of the real microbiology labs, there are a few, went out of business. The others are usually with the universities. The third one, procedural proxies. What otherwise is the realm of experts can now be evaluated, and rather handily, by work student studies with a crib sheet. Well, that sounds good, doesn't it? On to what they would measure. We already said organic matter, CO, HN, and the others. The unit is percent. You measure it through combustion. LOI is loss on ignition. LECO furnace is more accurate, but they're both okay. Don't take any high temperature, shortcuts and drive off any carbonate Cs. And for serious research, don't use Walkley Black. It's a chemical oxidation process. So now we're going to get into results. We're going to look at the soils from Silverbow Creek. That's called reclaimed. And then I had two controls, one riparian control and an upland control. Overall, the upland would be more relevant. Holy cow, we have a huge deficit. We're about 1/6 of the upland control, 1/8 of the riparian control. Bring in the compost truck. It's more amazing that the compost comes out of the hole somehow has about 0.4% organic matter. I can't explain why, but it does. We added 1.5%, that's upper 6 inches, and we ended up with 0.8. How'd that happen? Well, I think now you know. The labile stuff was oxidized and the CO2 went off, and the more recalcitrant stuff is left. The next test is readily oxidizable or active carbon, or you could call it permanganate oxidizable carbon. It's a proxy for the microbes that oxidize organic matter during growth, reproduction, and maintenance. It conveniently approximates the energy sources available to microbes. I couldn't tell you how good potassium permanganate is in degrading the same substrates as microbes. But it's the kind of thing you get from a proxy. It's, the units are parts per million carbon in dry soil, and you oxidize using a 0.02 molar potassium permanganate solution, and it's colorimetric. So let's look at results again. Now the reclaimed is 100, and the upland control is 160. So instead of being 6 times more, it's just 1.6 times as much. And there's more of a deficit with the riparian. But the important thing to see is what's active is much closer to a control in revegetated cover soils than the total organic matter. This one I think you're all familiar with.
The slides aren't being forwarded. The last slide I see is Betsy Dyer's quote, just so you know that.
Anyone else is having that problem, the same as her?
I don't
said
know.
the slides aren't advancing.
All I can say is they are on here.
Yeah, they are. Herb, is that you?
Yeah, this is Herb.
Yeah. Is anyone else having that same problem? Because it's showing that it's good on our end.
Yes, I don't see them either.
You don't see them?
Well, they haven't been advancing.
Are you still stuck on the same one, the Betsy quote?
It's
Oh my gosh.
Slide.
Right, we're seeing ratio.
Uh-huh.
Yeah, and I mean, I'm watching Zoom, and on our end, it shows that we're on the next slide.
Yeah. Everything on our end shows that it's advancing.
Do you see the
Yeah,
camera?
they're not advancing.
Okay. So it's just a few people that are having problems. My apologies, but
See there.
It might be your internet connection.
Oh,
Okay.
yes, I'm sorry about that. Kind of racing along here too.
You're doing good.
Well, we get back on target here. We were talking about proxies for measuring soil health and and how they're so attractive because any kind of a soil lab can do them. You don't have to be a microbiologist that's with a lot of experience to do this. It's much easier than that. And we were saying that the which one were we up to? Oh, the active carbon is closer into the reclaimed soil than the total amount of organic matter. And then we were going on to the C to N ratio, the carbon to nitrogen ratio, which I think everybody is familiar with. When it's wide, microorganisms are mobilizing soil nutrients, and whereas little nitrogen limits decomposition because it all has to come out in ratio. When narrow vascular plants get more nutrients, that's what we like is narrow. Other nutrients are more or less in ratio, and that means the ones that come from organic matter, not from minerals. So let's look at the results for this. Nitrogen usually comprises something like 1/20 of the soil organic matter, that's 5%. Something like 10 to 1's the equilibrium value. We look at reclaimed, 8.3. We look at upland, about 10, and riparian, 12. Well, since we like a narrow ratio, now reclamation has the advantage. It has the narrower ratio than the controls. Advantage goes to reclamation on that. Generally, a narrow C to N ratios are associated with high resource quality, with a vigorous active microflora, and favor mineralization. These are all things we like. Young succulent tissues are more easily decomposed than mature plants. So that means young revegetation is going to be better than an old stable plant community. Another test is burst respiration. Again, into some stuff that I like here. This is an actual measurement. Measures the metabolic activity of the soil microfauna. So it's fauna in this case. CO2 is measured at 0 and 24, in this case is 24 hours. Use the universal gas law, and you get parts per million of carbon dioxide carbon per gram dry soil. It measures the metabolic activity of the soil microfauna by the consumption of oxygen or the evolution of carbon dioxide, which is what was measured here. A link with active carbon is expected, but arsenic and metal contaminants can interfere. In brief, a soil sample is dried, sieved, weighed, tamped to approximate field capacity, and placed in a sealed container. Water is added, and right away, the CO2 concentration is measured. 24 hours later, the concentrate is again measured. Using the universal gas law, the results are calculated and reported in parts per million carbon dioxide C per gram dry soil. I like it. It's not a proxy, but a direct measurement of microbial metabolism. How do we do on that? Well, pretty good. Between the upland control and the riparian control. So reclamation is looking pretty good in some of these other measures. One more that I like, nitrogen mineralization. You measure nitrate when sampled, and 28 days later and divide by 28. It tells you how much mineral N is generated per day, another true measure. And we look at that and we say, okay, it's not as good as the upland control, but not too bad. However, this one has a really strong temporal relation. So what we're looking at here is the mineralization rate versus field age in years. We have an amazing coefficient of determination of 0.97. You know how rare that is in biology. And that shows you that in something like 25 years, this cover soil could equal the upland reference soil. And then I said might equal the riparian, but it won't equal the riparian because it's not a riparian setting for the most part. So while cover soils lag reference soils in all but C to N, but also, I should have said the burst respiration too is in between them. Positive temporal trends in soil health are evident. I conclude that soil function has been restored to a useful degree and is functional in the important sense of sustaining vegetation by cycling carbon and nutrients. Fresh exudates and plant root turnover from revegetation seem to provide enough labial C and nutrients to maintain soil health despite the lack of an OM buffer or reservoir in the soils. But I'm not saying that it's equal to a mature soil, but it certainly can function well. There's one other measure that I haven't mentioned, wet aggregate stability. It's a measure of the extent to which soil aggregates resist falling apart, which means slaking when wetted, and it's exposed to a force of raindrops. It's a physical property, also a biologic one. It's very sensitive to procedure. The NRCS office showed me what they do. You know, the word rinky-dink comes to mind. And my work was done by the Oregon State Soil Laboratory, and they use a Cornell rain machine to do this, which Cornell developed the procedure. Wasn't discriminating. I hope you get like that. Wasn't. The reclaimed is equal to the upland control. Really? Really? A couple years old soil has the same aggregate stability as a couple thousand-year-old soil? I didn't think too much of this one, so I didn't include it with the results. However, one of the soil scientists for a soils lab said, soils with high aggregate stability have less soil erosion, better equipment trafficability, I don't even know what that means, faster water infiltration, less surface crusting, and more diverse habitat for soil microorganisms. If I had to choose just one soil health test, I'd pick soil aggregate stability. To which I add, if I could just get one dude to promote my career, I would pick this one. So here it is. Fresh root exudates and plant root turnover seem to provide enough labile carbon and nutrients to build and maintain soil health, despite the lack of an OM buffer in cover soils. So the focus, sometimes the exclusive focus on total organic matter is inappropriate. This in turn abets nutrient cycling to sustain primary productivity. Here's a summary of it. I wish I had a pointer to show you. The one on the left is organic matter, huge deficit. This is the ratio of the reference soil to the cover soil, huge deficit. Nitrogen mineralization, okay, that's a pretty big deficit, but that's the one with the cool linear relationship. Active carbon, closer. Burst respiration, closer. C to N ratio, well, it looks like a deficit, but in this case, we like the low numbers more than the high numbers, so it's actually an advantage. And there's the wet aggregate stability. We're right back where we started. Primary productivity drives the soil food web. Nutrient cycling in turn sustains primary productivity. It's a tight cycle in which the larger recalcitrant OM reservoir plays a rather minor role. Revegetation abets cover soil genesis to true soil. That's what I'm seeing. When you have revegetation like this, it's both diverse and productive, I think we can assume that, yes, the soil is healthy. And now for the extra credit session, soil structure. I know there's some people that are interested in this. It has two components. The first is aggregation. It's the aggregation of primary soil particles, you know what that is, sand, silt, and clay, into aggregates or PEDs. I'm not going to use that terminology, PEDs. The aggregates are separated from adjoining aggregates by surfaces of weakness or voids. Here's something from a microbiologist. He says soil aggregates serve interest microbiologically because the cell material and excretions of bacteria, fungi, and actinomycetes are factors affecting the formation and stability of granules. What isn't structure? Well, it's not clods. It's not the stuff that you see when someone tills a field and slakes when it's wet. It's not fragments caused by the rupture of soil mass across a plane of weakness. There's always going to be a plane of weakness. And it's not concretions caused by cementing compounds. This is a golden sunlight cover soil. Every time I see it, I can't believe that we get good revegetation. And I took a picture of that every year. And one year I came back and you can see it cleaved in too, in the middle there. Okay, That's not soil structure, but it definitely shows the cleavage. This is a bunch of acidic soil that was limed with sugar lime. And the next year, we had this cementing into clods from the sugar lime, which was kind of upsetting at the time, and then it disappeared on its own. So it was more of a short-term thing. And this is what you get when you dig a hole, at least in kind of sandy material. The structure is massive, and yet it's going to cleave somewhere into smaller pieces. I wish I had an audience, but I was going to ask you a question here. This is an abandoned road. It's probably saline, that's why it's white. I don't know if you can see it, but you can see it puddles water too, and it's not naturally colonizing with plants. What kind of structure do you think that has? Well, I wish I could hear from you, but think about it. What kind of structure would you think it'd be under there? Well, there are 4 primary types of structure. There's platy. The particles are arranged around generally a horizontal plane. It's found in the E horizon. Trust me, we won't have any E horizons here today. The zone of greatest eluviation lost to leaching, you find it underneath conifers, for example. Platy structure is found. It can also be caused by compaction. So maybe there's platy structure under that picture of the road I showed you, caused by compaction. The most developed beds occur in the B horizon. That's the zone of accumulation, illuviation with an I. Calcium moves first, that deflocculates the soil, then clays to accumulate here over time. This is where you find angular blocky, subangular blocky, prismatic, and columnar structure. Prism-like, the particles arranged more or less vertically in columns and bounded by relatively flat surface. If the caps are round, it's one. We don't have to worry about that. One of them, I can't remember, prismatic, I think requires salinity. And then there's blocky. Beds are bounded by adjacent beds and shaped as if molded to fit among other blocky beds. Blocky has flat surfaces and sharp corners. Subangular beds are more rounded. Then there's spheroidal. Curved surfaces with little or no accommodation to surrounding. We won't worry about the difference between crumb and granular. They can be less than 1 millimeter or more than 10. Granular and crumb structure are most common in the A horizon, where organic matter accumulates. And I can't point to them, but this is from soil taxonomy. So the upper left are prismatic and columnar. And then the next two above each other, that's angular blocky. And to the right of that is subangular blocky. Next in the lower left is what platy looks like. And last is spheroidal. Let's go back up to D. Subangular blocky. Let's go to F. Spheroidal. Now, you hear single grain structure. That's really structureless. That's it's basically what you get in sand. Massive, however, refers to a lack of structure where every particle sticks to its neighbor. It's seen in C horizons of heavy soils and puddle soils. Well, that road I showed you could also be called a puddle soil, so maybe that had massive structure inhibiting percolation through it. This is an internet picture of massive structure. Just looking at what the stuff around the guy's feet there, I think that's probably a fairly recent fluvial deposit and no differentiation within the soil. This is one of my pictures. I hope you can look at that really intently. I see no structure here. Now, I'm sure there is some microaggregation. Remember, that goes on down to a 10,000th of an inch, okay? But that, to me, is as good a picture you're going to see of a soil that lacks visible structure. This would be an example of massive. That's on the Clark Fork River. And yet there are cleavage planes in it. And here's part of the problem. Soils without aggregation can settle and compact so that water is retained throughout the profile, blocking gas diffusion, potentially creating anaerobic condition. Root penetration is difficult in clays lacking structure. We don't have many clays here, but I put that in for the people that work with clay soils. Here you go, microaggregates. They are tiny, less than a 10,000th. You can't see it. They're bound by clay mineralogy and iron and aluminum oxides. Okay, still in the microaggregate from a 10,000th to a thousandth. What's a thousandth of an inch? Well, if you have good eyesight and you had a gap of a thousandth and you held it up to light, you would see it. And you could barely feel it. Or you can buy a set of feeler gauges. Sometimes they don't go down to one, but it's in that area. And they arise from the pressure of roots. So already at this small scale, plants are involved. Orienting clay particles face to face, plant microbial mucilage, and compression of local areas during drying. So already we have a role for plants, a role for microorganisms, and a physical process. Oh, here's some cementing agents. The point being here, there's no shortage usually of cementing agents, and silica is one too, which is even less limiting. Now, I didn't call this micro anymore. We're going from 1,000th to 100th of an inch. Aggregation results from encrustation by microbial residues around fragments or relatively recent, actively decomposing plant debris. This size requires continual input of plant matter, as do macroaggregates. The sticky envelope of prokaryotic cells, we know now that means bacteria, the glucocalyx, occurs in two states, a capsule and a water-soluble slime layer that enables cells to adhere to external surfaces. And that's going to also make aggregates. The big category, the stuff that we see most easily, 100th of an inch up to 3/8, are stabilized aggregates of primary particles or microaggregates. So there's more than one stage of aggregation. Fine and very fine plant roots and mycorrhizal hyphae are important structural components in this category. They must be continually replenished. So we're right back to we have to have a vigorous plant community. We have to have the microbial community. You get soil structure. This is a internet picture, somebody's idea of granular crumb structure. Pretty well defined, I would say. Another internet picture. Oh, I'm going to miss having a pointer here, but what I see here is, this is from one of the soils in the study here. Aside from the fact there's a couple rock fragments there that are coated with clay, I see aggregation of granular aggregation everywhere. And if you told me that you saw some subangular blocky, I would not argue with you. But we know that usually forms in the B horizon, but you have to go with what you see. I should tell you that I enlisted one of those soil scientists that I mentioned at the beginning, okay, to look at these photos with me, because there's nothing beats experience. So I wanted somebody that as I say, looked in 1,000 holes and knew what they saw. This is the riparian control. Don't look at that flat surface on the left. That's an artifact of the shovel, okay? But on the right side there, yes, I would say well-developed granular and quite possibly subangular block in there. This is to get our eyeballs calibrated. All right, what do we see here? Well, the thing in the middle of it is a little clod of sugar lime that was in the soil there. But if we look at this, and especially if you look above the sugar line, where the roots are the densest, and the roots are really dense in this, I can tell instantly myself that this Nevada bluegrass to provide all those fine roots, that has probably moderate granular structure, at least in the upper part where the roots are the most common. Here's another one. This time the white is not lime, but it's a crystal tuff, rhyolite tuff in there. And at best you could say for that, weak granular structure. That's a 6-year-old soil. This one also has that crystal tuff in it. It's a sand. It's a sand. I didn't expect it, but look in the upper left where it's most distinct there, and it's also shallowest with the most roots. You cannot say that that looks like that picture that didn't have any soil structure. So that has at least weak spheroidal structure. Here's another sand. Here you can really see the gradient. Look how much better developed the granular structure is at the top of the photo than farther down. So we're seeing it form pretty quickly, even in a sand. And a sand, by definition, has less than 5% clay. Okay, this is a tough one because levering it out with the shovel and do some cracks. But again, minimum moderate granular structure, and even in the upper part, maybe even strong. And again, if you see subangular blocky, I could say I see it too then. You have to go with what you see, no matter where it's supposed to be. What do you see in this? Well, I see a piece of wood compost after 16 years. It doesn't look like it did much good. What I'm really showing you here is you can't do structure for a wet soil. Here's another one that's been reefed out, but it's my oldest soil. And you can see compost in there too, if you get a good enough picture of it. 1.2% organic matter. Forget the big cracks, those are caused by me, but weak to moderate granular structure. Anytime you get a clay, you're going to get more structure. So that's one of the things that the small particles promote it. And here's a sandy loam, 3 years old. And I see granular structure pretty much throughout the whole thing, even after 3 years. So some of this structure could have maybe come in with the cover soil, but we scrape off the surface layer when we get the cover soil to get the weed seed off. And I really doubt it. I think that it actually formed in just these couple years. So what do we learn from this? Well, we don't see alluviation, that's lost by leaching, or illuviation, that's accumulation in the B horizon, nor do we see an utter lack of soil structure, except in that one photo. Granular structure is the most common by far, and it's found in the densest root zone. Tiny voids between aggregates are unconnected and don't provide aeration and water movement the way bigger macro aggregates and larger, better defined channels do. I guess I should have shown you a picture that showed a more developed structure of that type, but I don't happen to have any. When I did stuff like that, that was before digital photos. And clayey soils show more structure than sandy loams of the same age. Soils with more organic matter, big differences, not a couple tens, show more structure. We also see slightly more structure in the upper 2 inches than below that. And the caveat for this, I sampled the rhizosphere and not the bulk soil. None of the photos extend below 2 decimeters. And you saw the only clayey soil that I sampled. There it is, 15 years old, revegetation on Silverbow Creek. It has granular structure. The productivity is driving the soil food web. The soil food web is driving the primary productivity, and it's a healthy system. Thank you so much. I know that was a ton of information, but I was hoping that for the people that are looking for this, they would get a good overview. Any questions? Oh, is there still the old question?
Anyone have a question for Rich? Comment, good reminder that the soils are living.
What's that?
It's a good reminder that
Oh,
the soils
yes.
are living.
Yes.
And Rich's email address is also on the list of attendees. So if there's something that you think of after the fact, please feel free to reach out to him and ask him any questions you may have.
I hope that didn't go over everybody's head, but it's a subject that I became so interested in, monitoring vegetation. Really, what's the question? The question is long-term. Do we have a healthy system that's sustaining itself? And that leads you right into the soils.
Yeah, that was a great presentation.
All right, thank you.
Yep, thank you, Rich. Thank you for closing out the conference, and thank you very much to everyone who joined us for the last 3 days. It's been interesting, it's been different, but I think all in all, I think we've had a fairly successful event. Evaluations will go out. Professional development hours are available to people that are interested. Please let me know if you need a professional development certificate, out, indicating, the credits that you earned by attending. And I thank once again our sponsors. For without them, this event would not have been possible. I appreciate your support, and hopefully we can be all back together in Fairmont next year. So I will also get the e-gift cards out to those that won the Amazon gift cards. And if anyone has any questions, comments, concerns, please feel free to reach out to me. Oh, we actually do have a question for Rich. Okay, if there are numerous clays in the soil, what would be a good approach to get growth?
Well, you have hit on something that I have learned in my career. I also work for coal mines where a clay loam is your average, and and we had by violating the screening criteria, we had some clays also on the Silver Bow Creek, which were instantly recognizable by having poor revegetation. One of the main problems you have with the clay is you have overland flow. You can't get the water into the soil, so it's wasted in a sense. Off it goes. The thing, and seeding it is hard because you have to seed so close to the surface. But if you get your seed too close to the surface and then you have a windstorm, your seed exits. So coal reclamation is definitely harder to grow sagebrush in. They're very envious of me when I show them what I've done with a whirligig, a little cedar on my chest, and come up with these beautiful stands of sagebrush. And they're doing seeding this sagebrush twice, or putting it in the drill mix and putting it in the broadcast mix and all these things. So what can you do with a clay soil? Well, unfortunately, the first thing I can tell you is if you don't get good establishment in the first year, before you get weeds or anything to inhibit it, you should reseed instantly. And the other thing, and this is the good news for it, a lot of those clay soils that start out, let's say you want 1.2 plants per square foot. That's your minimum, something like that. And you say, well, we got 0.8. That's not so good, you got a lot of unstocked spots. Some of those, in a couple years, they're still receptive to falling seed. In other words, it hasn't sealed up yet. You have little cracks in the surface. And we've had some of them come out. You can't even believe it's the same site after 3 or 4 years. But they are definitely harder. When I went to Glen Rock and they were going to show us how to seed sagebrush and stuff, guess what? They had a sandy loam. We're all coming from mines that have clay loams. And they're making it look like it's child's play, but it's anything but child's play. I'm sorry, that's all I could tell you on that.
Okay, any more questions? Yeah, the professional development hours will be mailed out within the next couple of weeks. If you do want to receive a certificate, please send me an email and I will get that out to you. I also want to stress one final thought is, please look through your list of attendees and hopefully utilize the information as far as contact information that's available to do some networking with people who were a part of this event, specifically our sponsors and our speakers. So thank you to everyone who presented at the event, and I will be in touch with news for next year. Next year's event, we are booked for Fairmont for April 25th through the 29th, I believe. We're slightly earlier next year, but hopefully we'll be able to all be together at Fairmont. So thank you, and we will sign off for 2020.